My name is Birgit Schilling. I'm the president of US HUPO, and I'm very delighted to kick off this new group that we have. This is called New Frontiers in Spatial Omics. This is very special and very exciting. We have our co-chairs here today, Dr. Peggy Angel. She's an associate professor at the Medical University of Carolina in Charleston. And then also Kristen Burnham, and she's a senior scientist PNNL in the Pacific Northwest. So yeah, I couldn't be more happy to have you here and to learning a lot today. And well, thank you so much for initiating this. This will be a whole series. So I'm really looking forward. Peggy, do you want to get us started?
Sure. Thank you, Birgit. So I'm just going to share my screen here. And so can everybody see that as the main presentation?
Yes.
Yep. Great. So as Birgit said, Kristen and I are co-chairing these special seminars, and we're thrilled to do this. We've been doing imaging mass spectrometry for over 36 years between us. Kirsten as a graduate student in Caprioli's group and myself as a postdoc in Caprioli's group. And so one of the goals that I want to have with this series is I really want to educate and disseminate information about mass spectrometry information to an audience that's already doing omics biology by mass spectrometry. So we have a lot of members in our imaging community, and we want to introduce you to them so that you can learn about our resources. And there's a lot of people picking up mass spectrometry imaging, and so we're pleased to offer any assistance with that we can. So we've been through it all, from all the manual spraying to the technological developments that are currently present. And so that was one of the other things, is to understand what are the current technological advances and state-of-the-art mass spectrometry imaging, and then just increase awareness of resources and events. So our Mass Spectrometry Imaging Society in North America has hands-on workshops that are being hosted. So we invite you to visit the website. We do have an international mass spectrometry imaging conference happening next year, and we'll have over 500 researchers there who are using mass spectrometry imaging in particular for doing everything from metabolomics to trying to look at RNA by mass spectrometry. So it's a huge range of omics. So I'm going to start this session. Let's see here. So what I want to talk about is some fairly new technology that I've developed in my lab, and that's looking at extracellular matrix. So that's the stuff around the cell. And currently we're in the single cell era. So I just want to point out this particular advancement. So what I'm going to do is really talk about extracellular matrix in breast cancer. And as I go through the study, I want to discuss translational studies by multi-mass spectrometry imaging. So just to start out with breast cancer, so this is the second highest cancer type among women in the world, with over 2 million women affected by that. More than 500,000 women die from breast cancer just in last year, and this is kind of astounding given our modern era. So the main clinical issues here are diagnosis of risk. Can we stop cancer before it happens? Screening methods, and differentiating the breast cancer types for better treatment options. So when we look at breast cancer risk, this is typically clinically evaluated by mammography. And so here we look at an increase in breast density. And as the breast density increases, which is this white content seen in the breast, this is actually an increase in breast cancer. So it correlates significantly to breast cancer, and also allows clinicians to evaluate for tumor growth within the breast. So what's really happening in the breast is it's changing from this adipocyte tissue, which is a very lacy, fatty cells, to this very dense stroma containing breast. So this pink stuff here is actually the increase of collagen and extracellular matrix in the breast. So there's a direct connection between increasing the amount of extracellular matrix in the breast and breast cancer risk. So the challenge that happens with breast density is here you can see with a fatty breast, it's really easy to detect the breast tumor, whereas in a very dense breast, it's difficult to detect. And the other fact is that these tumors often arise from the more dense regions of the breast. So again, if we look at these histologically, which means we're looking at a thin tissue section that a pathologist would see, you're going to see this pink stuff as stroma. And you can see how it's spreading out into these fatty cells. So whenever we're doing mass spectrometry imaging, we're always connecting these to what is seen within the tissue. So it's not just breast density, but it's the fact that tumors grow from those and obscure the tumor growth. So what researchers have found in studies, translational studies and animal studies, is that it's the organization of collagen stroma in the breast that can actually predict breast cancer progression and also predict outcome. And so there's this very systematic change in collagen where it goes from this wavy pattern around the tumor, so this is the normal breast collagen density, to tangential to the tumor. And then when the tumor becomes metastatic, we have this swivel of collagen fibers going out from the tumor at a 90-degree angle. And so this whole process is so systematic that researchers like Conklin and Keeley have shown that you can predict breast cancer progression and outcome just from the organization of collagen around the tumor. And so what Patricia showed is that this tumor growth was actually caused by the breast cancer cells. I should say the collagen changes at the tumor margin were caused by cancer-associated fibroblasts. And so those had a very systematic arrangement that promotes tumor growth and metastasis. But how the collagen changes has remained largely unknown. And so we look at breast cancer across the continuum of cancer control. We know that this collagen stroma is involved in prevention and screening, diagnosis and survival. And then in therapy, what we look at is also looking at how this collagen arrangement changes when drug therapies are applied. So again, it's collagen organization. And in fact, if we look, in many different studies, it's qualitatively the alignment of collagen, the collagen type by transcriptomics, and then the post-translational modifications through prolyl hydroxylase that we're going to talk about in a minute that change. And what you'll notice is that none of these actually address the protein structure and how that's changed. So they're all at the qualitative level of looking at the collagen fibers, or they're looking at the transcriptomics, which, as we know from proteomics, there's timing difference, particularly for extracellular matrix. And so my lab actually focuses on the collagen structure by proteomics. And so we focus on post-translational modifications that involve proline hydroxylation. This is a very important modification that is involved in the triple helical structure. So this triple helical structure is held together through hydrogen bonding. This controls how this formation is accessed by cells. So this is integrins, which are involved in migration, attachment with the discoidin domain collagen receptor. So the open and closed facets of this triple helical actually help the cells know what activities that they're supposed to do. And the discoidin domain receptor is upstream of the many different stromal signaling paths, particularly the PI3K pathway. So this triple helical structure controls what the cells do and is also signaling responsive. So it changes how the cells think of things. So when you're thinking of the extracellular matrix, you want to also think of it as a three-dimensional construct that the cells are moving through. I always, do my hand waving here, but the cells are, it is a three-dimensional environment with a tight control of chemical gradients, and this structure is how it's controlled. Now, what we know from research in osteogenesis imperfecta and Ehlers-Danlos disease is that very small changes in this triple helical structure result in disease. So a single mutation from a glycine to an alanine, which is a methyl group, can result in disease. And so in our case, what has not been done yet is actually look at the changes that open or close this triple helical structure, the modification of this addition of hydroxyl group to the proline. And so this modification has requirements of using proline hydroxylases. There's 3 different enzymes. It is required to have oxygen present as well as ascorbic acid. So many of you might be familiar with scurvy. And so that's a disease where you have a lack of vitamin C, which was previously seen in days of sailing, with sailors having a lack of access to fresh fruit. Nowadays, you find it common in obese people who are also having an access to fresh fruit. But if we look at the FOSTA sequence, the amino acid sequence of the collagen structure, I've highlighted all the prolines here. So this center area, which I'll show again on the next slide, is the triple helical region of collagen. And the technical challenge here is that this triple helical region, which isn't just a linear formation, but it's twisted in association with collagen alpha 1, A1, and 1, A2. So it has very few lysines or arginines. So this isn't a very tryptic, accessible structure by conventional proteomics. And so what my lab has done, particularly to access and understand breast cancer, is use an unconventional enzyme, collagenase type 3. In this particular enzyme, it's actually a blend of enzymes, so there's other helper enzymes in there, and so this has a provisional patent on it. But this enzyme accesses this triple helical region, which I've outlined in blue here, and so it has a preference for GXP, or hydroxylated proline, which I represent as HYPE, or GPX. So this is the motif that it accesses by proteomics, so you can see a lot of glycines and prolines present. And particularly, we've developed this to be applicable to formalin-fixed paraffin-embedded tissue. So that's another step that is different from other techniques. And the reason being is that formalin-fixed paraffin-embedded tissue is available by the billions, literally, associated with clinical data. And so when we use this method, around 40 other ECM proteins are also reported. So a typical workflow will involve scanning by multi-imaging mass spectrometry. So that's matrix-assisted laser desorption ionization imaging. So there's many types of imaging, but today we're focusing on using multi-imaging mass spectrometry. So here we just use a laser to scan across the tissue. And at each X and Y coordinate, we produce a mass spectrum. Now, if we highlight very specific peaks, we can get a map of that peptide's expression across the tissue. So a typical workflow here with clinical samples is going to involve doing a hemotoxin, the eosin stain. This is a pathological stain as seen here. We spray the collagenase onto the tissue with a very dry sprayer so that everything remains localized. And we image by a mass spectrometer. In my case, I'm using a 7 Tesla FD-ICR. The TIMS Toflex is also another one. And then when we're finished, we also scrape the tissue and we can isolate tumors in different regions for LCMS-MS proteomics. And at the end, we have defined pathological regions that we can connect with identification and allow us to understand changes across the tissue. So I wanted to talk a little bit about sample preparation currently. So in the previous days, when I was a postdoc and Kristen was a student, we did manual spraying. And so it was very laborious, and it really depended literally if it was high humidity outside. It depended on the 3 hours that you would spend spraying one tissue section. So it was very challenging. So now we have automated sprayers that are very fast and highly reproducible. And so now biological variability is a primary source of variability for these studies. So in terms of instrumentation, the common laser for matrix-assisted laser desorption is a triple down Nd:YAG laser. And so these are now very highly refined lasers that can fire at up to 20 kilohertz. And combined with accurate stage movement, you can get around 20 pixels a second and go down to 5 micron imaging size. So you can really get down to a true single cell size. So the studies that I'll show today use a timsTOF flex and a MALDI FTICR. So both of these have dual sources so that we can use both electrospray ionization. We've done some proteomics on the timsTOF, and then we do a lot of imaging on the MALDI FTICR for access to high mass accurate data. So just going back to the workflow again, we have the pathological stain, we spray the enzyme with automated sprayers, we image it with fast instruments, and then we follow that up with sequencing information. At the end, I'm also going to talk about some other ways that we get sequence information from the tissue. So this is an example of collagen tissue imaging proteomics. So here we have a lot of images here. We can spatially cluster these. So when you do a heat map, you have generally some linear columns that show intensity of gene or proteomic expression. Here we have the spatial organization of those clusters. So each one of those clusters represents a proteome. So when we do LCM-SMS, we typically from this workflow get a huge range of collagens, but also other extracellular matrix. And so this is patient-specific stromal proteome in breast cancer. So we have fibrillar collagen and also end-linked modifications that happen on other extracellular matrix proteins. So this is a very specific method. So when we're targeting areas of stroma that is seen here in white, we get very high peak intensity. If we're off the stromal area here in the adipocytes, we get low peak intensity. So I just wanted to point that out. And then finally, one of the main points for this study is that we're actually looking at where this collagen structure is modified. So in this particular instance, we have higher probabilities of this particular proline being modified versus the other 3 prolines that are present in this sequence. And this particular sequence also connects with a cell attachment site on the collagen protein. So we're really mapping changes in the collagen domain as we look at the pathology associated with it. One of the things specific to MALDI imaging mass spectrometry experiments is that we can actually look at multiple patient samples at one time. These are done with tissue microarray. So a tissue section starts as this, we pull cores out, and we arrange them in an array. So from this, we get biological replicates, minimized batch effects, and we have a very single well-defined area that helps us identify target peaks of interest. So all of this, of course, takes a lot of teamwork. And so we typically do associate it with a variety of team members from clinicians to pathologists to statisticians for a study. So this is an example of looking at extracellular matrix in an FFPE block. And most of our imaging mass spectrometry also is associated with staining. So here we're using Picrocereus red. We've done our hierarchical clustering of the peptides that we see on tissue. And in this particular case, we identified a region that matched the tumor, so it clustered with the tumor. but it was next to the normal adjacent area that was defined by the pathologist for the core. So combining the staining with the mass spectrometry imaging, we went back and looked at this site, and indeed we saw a distal tumor as marked by these collagen fibers. And so this is an example of normal adjacent to tumor collagen fibers that would be seen in normal tissue. So you can see how they're kind of speckly. So using these combinations, we can actually associate collagen and extracellular matrix with exact sites of the tumor. So we can also look at changes in hydroxylation of proline distributed across the tissue. And so in this case, we're looking at collagen peptides without the modification and then peptides with the modification. And you can see that each one of those has a very specific spatial arrangement. Once we're finished with imaging, we take this tissue section and we scrape it. We can do the typical informatics that you would do by proteomics. And here we can see that not only we have collagens from this section, but we're also pulling down a number of extracellular matrix that are connected with regulating the collagen structure and hitting some key signaling components in breast cancer. Let me just go back to this slide. So this is where I think it's important for current techniques to also combine these cell signaling markers for these particular active nodes in breast cancer. So we have a combination of cell markers that we typically use. So this is a tissue microarray on breast cancer progression. I'm going a little faster now because I realize that I don't know how late we started, but I'm a little behind time. But so in this tissue microarray, which are commercially available, so if you're starting imaging, it's a great place to get some well-characterized tissue. So we have a range of normal, adjacent, non-cancer, and then cancer with different stages as seen up here. So we can rapidly scan this and compare it back to our tissue pathologies. And we can see that there are statistical differences now, as I've said before, in larger patient cohorts. So each of these cores represents a patient. So we can start understanding differences in breast cancer types. And then we can go back to our tissue section, which is called a lumpectomy, and we can actually look at how those gradient patterns correlate among the tissue microenvironment. So we've connected this also with microscopy techniques. This was done with Ed Brown at Rochester. And so again, it's the same tissue microarray, but now he's looking at collagen fibers by forward-backward scattering. So microscopy that's done on the pathology stain is seen here. And so what we've been able to find is that we have very specific correlations only in higher stages, which makes sense because that's when you have this very consistent change in the collagen fibers. But we have very consistent changes in parts of those structures that we can now connect with orientation of those collagen fibers. So the last part of my talk, I'm going to just talk about breast cancer disparities. So the topic that I focus on is looking at changes relative to ancestry, and that's because compare between Black women and white women, although there's the same incidence of breast cancer, Black women have a 36% higher mortality rate because of more aggressive breast cancers. And so they have higher stages and higher grades, over 2 times the occurrence of the triple negative breast cancer that has no great therapies occur in Black women. And so the factors driving this remain unknown. And so what we're looking at, we're looking at this association of collagen density, the immune environment, and fibroblast activation. So what we know is that geographic origins across the world, actually, and there's many different groups that we're looking at, have caused different unique and immune and inflammatory responses. And so this results in different tissue microenvironments, and we believe that this is the cause behind disparities in different types of cancer. So again, we're looking at collagen density changes as well as immune immune changes in expression pattern. And so when we look at these by ancestry, what we find is there's very specific parts of the collagen structure that are modified. And again, it's very specific prolines that we're marking as modified based on ancestry. So this is a quantitative proteomic experiment. So in this particular data set, we can associate the transcriptomics with this proteomics. And what we see is that we know we have prolyl 4 hydroxylase 3 upregulated in breast cancer. That's not a surprise, but what is actually different are these hydroxylated proline codes that are changing in these more aggressive breast cancers seen in Black women. So this is just some MS shown to show to the mass spectrometry audience. We get nice sequencing information from these, as well as nice localization of which prolines are hydroxylated. This is on a entire tissue section, and so this looks at the peak area across the entire tissue section, but we can also zoom in and look at particular features of a tumor. So likewise here, this is the same cancer genome atlas breast tissue that was shown in the proteomics, and we can look at peak intensity across the entire tissue, but also look at our spectral signal here, and you can see that there are clearly peak differences between African American and European American. And these are associated with the different sites of proline hydroxylation. I don't think it's just going to be ancestry based. I think women who are, more at risk are going to see these, and that's what we're working on totally in my lab. So this is a combined image. We can look at all these by imaging mass spectrometry, and you can actually pick out the tumor margins here, as well as some punctate signaling seen here in the European American invasive ductal carcinoma. So combined, this gives us a lot of information about the environment. But what's really important to us is to connect it with a cell phenotype. So my student, Denise Rajanarong, has developed a method where on the same section, we can do multiple immunohistochemistry, and we can actually map the collagen peptides to that. So we can connect that with that this proline is not modified and this proline is modified. So now we can get the enzyme staining done, and we know exactly where that is happening and connect that with the peptide modifications occurring in tissue. So here's an example with the mass spectrometry imaging data. We can now typically see both prolyl isomerases. So we can see the non-hydroxylated and the hydroxylated version, and we can actually overlay those on top of each other as well as quantify between sections. So this is just another zoom in. We're also looking at this in triple negative breast cancer, and we've seen very dramatic differences. Again, what you'll notice is I'm following the same portion of the collagen region here, where we know this proline is hydroxylated. So far, it's never been that proline during the progression of breast cancer, but also in the different tumor microenvironments seen here in triple negative breast cancer. So using tissue microarrays, we can go through and look at tumor changes. We can look at normal adjacent, which could be under the control of the cancer field, as well as normal breast. And so what we find is that there's distinct changes in the tumor microenvironment surrounding this height variation in this particular GPA, GER, GSP peptide, but this is inversely correlated in healthy tissue. So this is healthy tissue without cancer. And so we think that there's certain lack of modifications that can actually produce a permissive environment. So we've gone through with these same TMAs and connected them with looking at fiber thickness. This is just an example, and it wasn't the peptide that I've shown before, but looking at a particular peptide now among many patients and comparing between Black women and white women, and we can find peptides that correlate. So our next thing is to take that peptide and map it more closely to those collagen fibers. So in invasive ductal carcinoma, this is now not ancestry mapped. We've also seen the emergence of this GPA collagen peptide. And so here we're looking at invasive ductal carcinoma versus ductal carcinoma in situ, which is a disease that could progress to cancer. And what we think is that this particular peptide is present in normal, but it's more associated with reactive stroma. So one question that, and we've also looked at many other peptides and connected those with ancestry differences. And this is done by Taylor Houlihan, a student in my lab. She's also marking out peptides connecting to progressors versus non-progressors in ductal carcinoma in situ. So I wanted to finish up by talking a little bit more about identification, and I'm going to have to speed through this, but this is the example of the peptide that we were seeing before in breast cancer. We've gone back in many studies and done targeted proteomics to pull these down. So this is actually unpublished data on a publication that we had continued to pursue. So this is LCMS done on a whole tissue section, but then the identification applied back to including TMA. So you immediately see that it's not perfect, especially when you have many potential sites for proline modification. So what we've taken to do is creating synthesized peptides, identifying unique fragments, doing MS-MS on tissue to connect these to the disease-specific variants. And so this is just the fragmentation around this particular peptide. And so what we have done in this article that was published in JASMUS is we've gone through with synthetic peptides now, and we've mapped these unique sites that we can see. And you can see by eye mobility, there's not too much difference in eye mobility, but you know, it's enough to at least make some further connections with the collision cross section and that particular peptide. So my student Casey Clift went through and she actually targeted these areas manually to get MS-MS. And so what she found by comparing those back to these synthetic peptides by both eye mobility and fragmentation is at least the two main proteoisomerases that were present were these P1 and P2 and P3, but not these where both of these were hydroxylate, which kind of makes sense from a structural point of view. The problem is that we don't have exact spatial information. And so here there are some strategies for doing MS-MS imaging, which we'll have another invited group to talk about that later in the series. So the main points that I want you to take away is that enzymes present a tool for targeted access, and so that's what I've leveraged in my study. We use combination of enzyme staining, immunohistochemistry, and microscopy on the same tissue section to understand the biology. We have changes in hydroxylated collagen that may contribute to differences in ancestry, but definitely are involved in changes in breast cancer pathology. And then the height peptide identification is certainly there's a lot of room for advancement. We can get the main proteoisomerases by LCMS experiments, and then use ion mobility and MS imaging later on to localize these two specific cell types. So I'd like to thank my lab who funnily made all these little halos for us to wear and got together a group picture this summer. And then funding and patients and donors, as well as a really great collaboratory network. So what I'm going to do now is turn this over to Kristen Vernon to chat. And then at the end of this, we're going to have a brief discussion about the work and answer any questions. So Kristen?
Yep, great talk, Peggy. Okay, share that. And then, there we go. Does it look good?
Yes.
Okay, so as Peggy said, so I'm Kristen Burnham-Johnson, and I'm calling in from Pacific Northwest National Lab today, also known as PNNL. And for my talk, I'm going to be focusing a lot on bottom-up proteomics imaging, so LC, MS, MS. And like Peggy, I really want to emphasize the importance of imaging mass spectrometry experiments. So they're powerful because they can provide information on the spatial distribution of proteins across thin tissue sections, as we know. And this really allows insight into analyte co-localization and also the molecular basis of tissue features. So here we can see an optical image of a thin tissue section, which is 12 microns thick. The grid pattern of each of those boxes that you see is 100 microns tall and 100 microns wide. And we can see that this tissue section is heterogeneous and is composed of multiple different cell types. We can see those cell types here. They're represented with false coloring. So we have stroma, we have epithelial cells, both glandular epithelium and luminal epithelium. But with imaging mass spectrometry, we really want to go beyond this. So we want to understand more than just cell type. And I really like this figure here. So with imaging mass spectrometry, our goal is to understand the molecular composition of tissues. And the focus of my presentation is proteomic imaging. So we want to see how proteins change, not just in cell type, but instead across the tissue microenvironment. And that's what we're depicting on this slide. So we're visualizing our proteins as gears, which are working together across the tissue microenvironment. So across space, across time. Therefore, we're moving beyond histology to start to understand that function that we want. So what are some imaging mass spectrometry approaches for mapping proteins? We'll depict it on this slide on the left as really nicely described by Peggy. There's MALDI, so matrix-assisted laser desorption ionization platform. So we have MALDI-TOF, MALDI FTICR. And on the right, we have liquid extraction surface analyses depicted here like LISA, electrospray ionization. Now, workflows for these imaging experiments, they benefit from simple setups. But there's caveats and limitations as with any type of technology. And we all know that tissue sections are complex. Therefore, there can be challenges associated with these direct measurements. Now, applying advanced technologies can help a lot. Two are depicted on this slide. So on the left, we have FTICR, Fourier transform ion cyclotron resonance for MALDI. And on the right, we have a FAMES Pro device for LISA. So these help improve signal and noise and extend proteome coverage, but there's still challenges. So what are the pros and cons of direct ionization for protein imaging mass spectrometry? Pros in green, simple setup, it's robust, it's high throughput, you get fantastic data. Cons in red, you can have effects like ionization suppression, which can bias measurements. When you think of proteins, there's over 40 million proteins per cell, and that's a lot. Therefore, we can have issues with low extraction and ionization efficiency. And also, these measurements lack the simultaneous protein identification information. So you have a mass to charge value and not a protein identification, and you can use secondary experiments to figure out what it is. So what I'm going to present in these 30 or so minutes is bottom-up proteomic workflows for mass spectrometry imaging. So in our mass spectrometry imaging approach, we're not directly ionizing proteins from tissue sections. Instead, we're leveraging this depicted workflow of sample preparation, separation, mass spectrometry, and data analysis shown in the 4 colored panels to really give us deep proteome coverage. So in this bottom-up proteomic workflow, a crude protein extract is enzymatically digested into peptides. Peptides are then separated based on chemical or physical properties prior to mass spectrometry detection and identification. And in our current workflow, we're using liquid chromatography separations. Now on the right, I have listed 6 advantages of our bottom-up proteomic workflow. So we can use the next few slides to really dig into those. So if we look at our sample preparation in detail on the right, we take our thin tissue sections and utilize phospholipase removal and lysis steps for efficient protein extraction and digestion into a peptide mixture. Therefore, our first two benefits are we have highly efficient protein extraction, and number two, peptides have high ionization efficiency. Now let's discuss our separation steps. We're currently using liquid chromatography separations to reduce spectral complexity. You can see in the chromatogram to the right here, all these peaks, which there are many of, they elute over time. Therefore, liquid chromatography separations, they reduce the spectral complexity and minimize the ionization suppression. And in this workflow, peptides are confidently identified by MSMS or tandem mass spectrometry. In the mass spectrometer, as you all know, we have our data-dependent acquisition, which utilizes cycles of full MS, followed by MSMS or fragmentation of the most intense peaks. So not only do you get that confident relative quantification that we want, but you also get the peptide identification, which can have a controlled false discovery rate to be less than 1%. Now, a similar idea was previously published by PNNL way back in 2007. So we've been working on this for a while. And in the study, a mouse brain was voxelated into 1 millimeter cubes. Sample processing of the cubes included homogenization and efficient protein extraction. Proteins were digested by trypsin, and then the resulting peptides were analyzed by liquid chromatography or a LC gradient coupled to FTICR mass spectrometers. But a reference database or a reference peptide database was used to identify peptides based on the LC retention time and mass. So we did not have real-time MSMS, and we only identified around 1,000 proteins from these larger 1 millimeter by 1 millimeter by 1 millimeter cubes. So in PNNL's new bottom-up proteomic imaging workflow, we're achieving higher resolution by a magnitude and better proteome coverage by over double, while also making peptide identifications in real time. So let's dig into the details of our new imaging approach. So it takes advantage of a bottom-up proteomic workflow to image thousands of proteins from tissue sections at better than 100 micron spatial resolution. Now, we've applied this workflow to study diverse human and environmental samples, but for this particular presentation, I'm going to dig into our first proof of concept 2020 manuscript, which is denoted at the bottom of this slide. Now, of course, bottom-up proteomics, it's nothing new. So what is actually novel about our approach? Well, the novelty of our approach is illustrated in step 2 of this slide. So our novel nanoscale sample preparation, which was implemented by first authors Paul Piechowski and Ying Zhu. So step 1 uses standard laser capture microdissection of tissue sections. And step 3 uses our traditional liquid chromatography separations, mass spectrometry data analysis approaches for peptide analysis and identification. So why does scaling down our sample preparation to nanoliter volumes for step 2 enable us to both identify and quantify over 2,000 proteins from our small samples, which again are our voxels only measuring 100 microns wide, 100 microns high, and 12 microns thick? And the answer is by miniaturizing our sample prep, we're decreasing analyte losses and also improving digestion kinetics. So first, let's discuss peptide losses in traditional bottom-up proteomic sample preparation. We take our tissue, we homogenize and extract the proteins. Then we denature, reduce, alkylate, and digest our proteins. We then desalt our sample so it's compatible with our mass spectrometry analyses. Now, during each one of these steps, as you can see here, we have losses due to surfaces and transfer steps. So all of which increase the overall sample loss due to nonspecific binding. So this will not work well with small samples. You'll essentially lose your proteins to surfaces and transfers. And besides losses due to surface exposures and transfers, you also have to think about the enzymatic digestion. For example, if we have trypsin digestion, it's inefficient when protein concentrations are low. So using traditional bottom-up workflows for small samples like our tissue voxels is going to lead to poor digestion efficiency, which is depicted in this picture here. So now let's dig into the fun part. Let's discuss our novel nanoscale sample preparation, which has been done in many labs. In our particular lab, we're focusing on nanopots. So nanopot stands for nanodroplet processing in one pot for trace samples. It was developed at PNNL by Ying Zhu and Ryan Kelly. Ying is pictured here, and their first publication on nanopots was published in this 2018 manuscript. So with nanopots, all the sample preparation steps required for a bottom-up proteomic workflow are performed in a nanowell with a 200 nanoliter reaction volume. So the picture on the left, you can see nanowells with 200 nanoliter droplets of colored liquid. The video on the right, which is just looping around, you can see the automated dispensing system, which fills and extracts liquid in the nanowells. Therefore, we have a robust way to perform all the proteomic processing steps, such as reduction or extraction, reduction, alkylation, enzymatic digestion with lysine and trypsin, and cleanup in 200 nanoliter volumes. And if we look at the table in the bottom right, you can see that the tube method versus our nanowell method, we have decreased our reaction volume from 100 microliters to 200 nanoliters. And we have reduced our surfaces by 99.6%, thereby decreasing sample losses due to non-specific binding, and we've increased our digestion kinetics by over 500 times. So in our proof of concept nanopots proteomic imaging experiment, we utilized uterine cross-sections from pregnant mice prior to the adhesion of early embryos to the uterine wall. And this was done in collaboration with S.K. Day, who is Director of Reproductive Biology at Cincinnati Children's Hospital. And if we look at the mouse uterine cross-section on the left, you can see that this is a small structure, not surprisingly, and the mouse uterus is only approximately 1 millimeter in diameter. Yet the tissue is heterogeneous, like so many tissues, with many different microenvironments, which is why we have previously used this system to test other MALDI and nanodesi imaging mass spectrometry approaches. So if you look at the false colored image in the center, you can see that the tissue section is heterogeneous, as I mentioned, and it's comprised of stroma cells depicted in pink, glandular epithelial cells depicted in green, and luminal epithelial cells depicted in blue. Now, the blue luminal epithelial cells, they line the uterine cavity that the embryo travels through, and there's essentially a molecular crosstalk between the embryo and the uterus. And this is what enables the embryo to attach to the luminal epithelial cells and then subsequently invade into the stroma, which is shown in the cartoon to the right. So therefore, by attaining molecular level understanding of this process, it informs new approaches to improve the outcome of natural pregnancy and also in vitro fertilization. So this was one of the sections imaged in our manuscript. Our tissue thickness was 12 microns and our analyzed or imaged pixels or voxels were 100 microns by 100 microns, which means we had between 30 to 60 cells in each pixel. So each pixel had over 2,000 proteins identified, and these pixels contain the different cell types, as you saw on the previous slide, including stroma in pink, glandular epithelium in green, and luminal epithelium in blue. So again, we can voxelate our sample section into pieces measuring 100 microns long, 100 microns wide, 12 microns thick, and visualize how over 2,300 proteins are changing across the heterogeneous tissue landscape. So this slide depicts our entire proteomic imaging workflow. In panel 1, we outline our area of interest. In panel 2, our thin tissue section is voxelated into pixels, which are collected into nanowells. If you look at panel 2, it's hard to tell from this, but our tissue is actually sitting on top of the glass slide. And so the voxel of interest is cut out with a laser beam from underneath. And once that laser has liberated the voxel, that capsular droplet, which is filled with a non-volatile solvent, DMSO in this case, is moved right above that liberated voxel. So the droplet's hanging upside down. The next step is to get the voxel into the droplet. That glass slide, it has a PEN membrane coating. So laser energy is applied to it. It constricts the coating. And by constricting the membrane, it actually propels that liberated voxel up into the upside down well. So a picture of the sample voxel in a nanowell is shown in the bottom left. And all the proteomic processing occurs in the nanowells, leading to that high peptide recovery due to decreased surface area losses, and also increased enzyme kinetics. And our mass spectrometry analysis, it utilized improved automated nanoliquid chromatography platform, which enabled full sample injection. And then we also developed our own software so that we could visualize the expression of each protein across the entire tissue. So data visualization is very important for imaging experiments, which produce, as you can imagine, a lot of data that needs to be mapped and visualized in 2D. So for our software, we utilize Trelliscope for the online data visualization, and Lisa Brammer led these efforts. So Trelliscope displays our data either at the protein or peptide level, where red is high relative abundance and green is low relative abundance. And as you can see by the scale bar in the center here, red high, green low. So this particular protein has high relative abundance in the luminal epithelium. You can see the red pictures, they match that localization to the luminal epithelium in the optical image, which is located in the center of the slide. And you can visualize all 2,298 protein images from this section within the Trelliscope interface. So let's dig into a few images. So these are some of the proteins with the highest relative abundance in the luminal epithelium compared to the stroma. You can see again that the high intensity red pixels matching the luminal epithelium cells in the optical image. So nice co-localization there. And to support our images, we use statistics. So as we all know, statistics and biological replicates are important for any study. So we have both of them with this. Details on the statistical analysis are shown at the bottom of the slide, labeled as cell type data. So we had 5 stroma, 5 luminal epithelium, 5 glandular epithelium samples, where we had 100 to 200 nanograms of these unique cell populations. They were captured from 3 to 5 sections for 15 samples. So the important point here, our proteins of interest are significant with an adjusted p-value of less than 0.05 in our cell type data, and have complementary spatial distributions in our imaging data. And these 4 example proteins have images localizing to the luminal epithelium, which is supported by the statistical data represented by the box plots, where protein abundances were significantly higher in the luminal epithelium, listed as LE there, compared to the stroma represented as S. So, I know there's a lot going on this slide. So the take-home point is we have high confidence in our imaging results. We use numerous biological replicates and statistics to support these findings. So an important goal of this study was to characterize the unique proteomic landscape of luminal epithelium cells. So these are the first cells to attract and make direct contact with the embryo. And in our study, we had 149 proteins that were significantly enriched in the luminal epithelium. So these proteins were significant with an adjusted p-value of less than 0.05 in our cell type data, and had a complementary spatial distribution in our imaging data. I've chosen 11 images here just to show as examples. And you can again see that high intensity red voxels localizing to the luminal epithelium. And then the top left of this figure is the top 6 luminal epithelium functional categories. So images of proteins localizing to the luminal epithelium. They had functional roles molecularly linked to epithelial cell crypt formation such as actin cytoskeleton remodeling, cell polarization, and cell migration. And all of these are really important functions for successful early pregnancy. So now let's switch for the stroma cells. So stroma cells, which are the cells surrounding the luminal epithelium, they support embryo growth during early pregnancy. And in our study, we had 175 proteins that were significantly enriched in the stroma. So these proteins were significant, again, by our adjusted p-value of less than 0.05 in our cell type data, and had complementary spatial distribution in our imaging data. We have 11 images here, just shown as examples, and you can see that high intensity red voxels are now localizing to the stroma. And in the top left of this figure, you can see the top 5 stromal functional categories. So images of proteins localizing to the stroma, they had functional roles molecularly linked to many things, including remodeling of the extracellular matrix, which is heavily enriched in the stroma region. And I want to slow down for 1 second here because I want to make an important point. So our protein images were able to characterize unique tissue microenvironments within the same cell population by visualizing the gradient expression increase of stroma proteins along that top to bottom axis of the uterus. And the reason for this is there's an invisible oxygen gradient that causes this expression change. So although these stroma voxels look the exact same by histology alone, our imaging data was able to detect this important change in the microenvironments of the uterus. So it's really showing how important imaging is for truly understanding function. So bottom-up proteomic platforms, they can dissect the complexity of heterogeneous biological samples. We had over 2,000 proteins that can be identified at less than 100 micron spatial resolution on thin sample sections. And this platform is reproducible as illustrated by the 2 experiments with complementary results on this side. And detailed spatial maps of proteins, lipids, and metabolites, they can be obtained by combining bottom-up proteomic imaging with other complementary imaging mass spectrometry technologies such as MALDI and nanodesi, just to name a few. So to expand on that last point, in addition to visualizing tissue type specific expression with our protein images, we were also able to visualize region specific bioactivity. So when we were processing tissues, we were collaborating with Ingela's lab at the time to advance nanodesi's ability to image prostaglandins. So I sent mouse uterine sections from PNNL to her lab at Uppsala in Sweden. And denoted on the right of the slide is her resulting publication. So the mouse uterine tissues for these protein images and these prostaglandin images were from the exact same mouse uterus. Now, our protein images on the left side, they were able to visualize the metabolism of arachidonic acid into bioactive lipid mediators, where prostaglandin GH synthase 1 and prostaglandin E synthase 2, they localized to the luminal epithelium. And to support our findings, so we could see the entire pathway, nanodesi confirmed that prostaglandins, including prostaglandin E2, they do localize to the luminal epithelium in the uterine sections here during early pregnancy. So we were able to visualize region-specific arachidonic acid metabolism and activity by combining results from multiple MS imaging platforms. So as I mentioned in the beginning of the presentation, all technologies have pros and cons. So what are our limitations? Well, we have a problem with throughput. So our goal is to overcome the throughput limitations of our liquid chromatography mass spec based proteomics setup. So in this slide, we have 3 LC gradients options. We have 30 minutes, 60 minutes, and 120 minutes. And not surprisingly, we obtained the most peptide identifications by utilizing a 120-minute liquid chromatography gradient. But the problem is with that long gradient, you can only run a max of 10 pixels or voxels per day. We could shorten the gradient and use a 60 or 30 minute to increase throughput, but then we're just decreasing the number of peptide identifications. So how can we improve throughput without decreasing our peptide identifications? Well, we're incorporating isobaric tags and sample multiplexing into a new workflow, which we're calling PINT, so proteome imaging with nanopods and TMT. I'll go over this in more detail in the next slides, but as an overview, we're using tandem mass tags, which many of you are familiar with, but this is to uniquely label peptides in different pixels. We then combine our peptides from different wells, which increases throughput while also decreasing pixel size. So what are tandem mass tags? Well, a TMT templex is an isotope-based high throughput protein analysis reagent. Structures for the 10 isobaric tags are depicted in the lower left here. So TMT labeling is an approach developed for multiplex identification and quantification of proteins from multiple different samples into a single LC-MS analysis or run. So because we have 10 different samples that we're combining into a single MS analysis, our proteome identification sensitivity is greatly improved, which enables mapping of much smaller tissue voxels without compromising the proteome coverage. So in a traditional TMT experiment, from each sample, proteins are extracted, the peptides are digested, then each peptide is labeled with the unique TMT tag. The samples are combined, fractionated, cleaned up, and analyzed by LC-MS. And then in the MS spectrum, which you can see at the bottom right of this slide, in addition to the fragment ions for peptide identification, there are also reporter ions in the low mass region, which are unique for each TMT tag. So enables quantification and identification from the same spectrum. So what does the TMT workflow look like for our PINT experiments? Well, we again use laser capture microdissection in nanopods to collect our luminal epithelium voxels and stroma voxels from a 12 micron thick mouse uterine section. Now our proteomic processing bottom-up workflow, it includes the tissue collection into our non-volatile droplet on the nanopod's chip, our protein extraction, and our trypsin digestion. But now, following that digestion, we label each peptide sample with a TMT reagent, and then we combine these samples for LC-MS analysis. And we also add 5 nanograms protein digest label as a reference, so all peptides can be compared even across multiple experiments. So that's really great. You can go to larger experiments this way. And since we combine samples, we now have more peptides with 13 nanograms total, which enabled us to decrease our voxel size to 50 microns by 50 microns by 12 microns. So if we compare the proteome coverage from our TMT and label-free experiments, we can see that the number of proteins identified are comparable, both over 2,000. Yet with the PINT experiments, we cut the voxel size in half, giving us a 50 by 50 micron lateral resolution. And importantly, we had 3 LC-MS runs instead of 24, which means our total MS time went from 60 hours to 7.5 hours. So a significant difference. So PINT can provide better spatial resolution, total less MS time, while still giving you over 2,000 protein identifications. And just to show you 2 examples here, so we found that the preliminary PINT images correlated with our validated label-free images. In these images, we did switch the colors a little bit. So red's still high intensity, but now blues are low relative intensity. And the luminal epithelium is outlined in yellow in both of our sections. So our label-free image is on the left and our PINT image is on the right. And you can see that vimentin has lower blue abundance in the luminal epithelial cells and higher abundance in the stroma cells in both experiments. And for my last example here, we're looking at images of the protein LOX15, which is a arachidonic acid processing enzyme. And as I discussed previously, we know this protein and its activity is enriched in the luminal epithelial cells. And you can see that LOX15 has higher red abundance in the luminal epithelial cells and lower blue abundance in the stroma in both experiments. And an important point here is that since the pint approach uses smaller 50 by 50 micron voxel size, you can see that the resulting pixels are more representative of pure epithelial cell expression. So in conclusion, our label-free or LCM nanopots tandem mass spectrometry approach, it provides a new way to visualize protein expression in tissue sections, including tandem mass tags for paint. It improves both spatial resolution and throughput. And both of these provide novel approaches for both biomedical and environmental research, which enables new research directions. Spatial measurements are important for everything. And for the acknowledgements, I want to thank Paul, Ying, and Ryan for all their help developing these new imaging approaches using nanoscale technologies. And thanks to Lisa for all of her work developing Trelliscope, so we had a way to visualize these large data sets. For funding, it was through NIH, through HubMap, the Human Biomolecular Atlas program, and also NICHD, which is why our proof of concept experiments were in mouse uterine tissue. And then we're located at an environmental molecular sciences laboratory. It's a DOE Office of Science user facility. So really by developing these new tools, we can make them accessible to the wider research community. And with that, I'll stop sharing so that we can take some questions.
Thank you so much, both Peggy and Kristen. This was incredibly inspiring and such beautiful data that you showed us today. We already have some questions, and I would like to encourage everybody to put their questions into the question and answer section. So maybe I'll start with the first question to you, Peggy. So Charles Schumann, he's asking, Peggy, you mentioned that your imaging works well on FFPE sections. Does the paraffin present a challenge, or do you remove it prior to imaging? Would it be better to prepare frozen tissue sections if we have the ability to prepare our own samples?
All right, thank you, Charles. That's a really great question. Most of the clinical tissues are embedded in paraffin, which is wax, which preserves the protein structure. So to get rid of it, and I'm going to share my screen a little bit. So to get rid of it, we basically reverse how the wax got there in the first place, and we go through a series of washes. And so this is exactly following the workflows that are used for immunohistochemistry. So our workflows for mass spectrometry imaging are very parallel with immunohistochemistry. And we use xylenes, ethanols, and water to remove the wax so that we now have access to this protein structure. And if you were doing an antibody stain, you would do the same exact thing. So one resource we use is IHC World. And so that has a lot of information on doing these steps such as dewaxing. For mass spectrometry imaging, particularly for tissue microarrays, I like to add a chloroform-based wash between the ethanols to get rid of the wax. You start thinking about details like laminar shear when you pull the slides in and out of this Copeland jar here. And so adding this chloroform-based solvent really is kind of a finishing move on making sure that those waxes are gone, and that's useful for mass spectrometry imaging. It's not used very much in immunohistochemistry, but the same wash, if you're going to do fresh frozen, which we've also done the same method on fresh frozen, you want to use this wash. Let me see if I can navigate to it here. So if you're accessing the protein structure, to remove the metabolites and fatty acids. And so this is one way we also stack our imaging experiments. So you can image the metabolites, lipids, whatever first, and then you want to remove those from the same tissue section because they cause ion suppression in fresh frozen tissues, so that you can then start accessing and seeing the analytes that are produced by an enzymatic digestion.
Oh, thank you so much, Peggy. Well, that's actually quite interesting. So you could even do multi-omics on the same tissues.
Oh, definitely.
That's
Yes,
amazing. That's super cool. Great. There was another question from Jacqueline Dunn, and this is for Kristen, you. So here the question is, for nanopods, is there any issues or controls for solvent evaporation? Really cool tech.
Great question. So yes, so we try to use non-volatile solvents like DMSO for our droplets, and then also for processing, it's in a humidified chamber so that you're not dealing with those losses. I know a lot of people wanted to do multi-analyte extraction with nanopots, get lipids, metabolites, and then proteins out. What I personally do in our lab for my experiments is I prefer to do nanopots for protein, and then I rely on MALDI and other spatial techniques like nanodesi for metabolites and lipids, because it's just really hard to beat the power of MALDI for those, and so they can kind of be combined. Because if we tried to do metabolites with nanopots, dealing with that small a volume and trying to use a volatile solvent for extraction, it's going to be difficult. So yeah, combine all the spatial measurements together and get the most out of it.
Cool. Yeah, so maybe I had another question also for Peggy. So Peggy, as you have sprayed the matrix on, and that was really nice what you mentioned, that this is now so automated, right, and so controlled. So once you have sprayed, how long is the tissue able to be measured? And also, have you done certain things where maybe you do a first round of imaging and all of a sudden you get another idea and you want to image a few other analytes? Is it possible to go back or re-spray, or could you comment briefly?
Yeah, it depends on what your new idea is. So typically in our lab, we do glycomic imaging first. So we look at N-glycans. And
Right.
so in that case, it's helpful for proteomics. It's very similar to proteomic workflow where we're removing the glycan sugars. In our cases, most of our tissues are very rare and valuable. So we just have to look at this metabolic post-translational modification. But once we finish with that, we can do a number of different enzymatic treatments to look at the protein structure. So we can look at collagen imaging, we can look at elastin imaging, we can look at tryptic imaging. So we can just do that in a series. You do have to be careful because sometimes going through all of those workflows can be pretty tough on the tissue. So if your tissue's robust, like hepatocellular carcinoma sections will hold up well to this, then you can process it through that whole, many different tiers of enzymatic imaging workflows. If you're in fresh frozen now, you can actually start at the metabolomics and work your way up as long as the tissue stays on the slide.
Cool. That's so inspiring. Yeah, thank you so much. And for everybody, we have recorded this webinar and it will be on the US HUPO website. And also, I'm really excited. This is the start of, number one of a series of talks by Kristen and Peggy. And I'm excited to announce our next meeting will actually be on Friday, November the 11th. And we'll also have a workshop at US HUPO, which will be in March in Chicago. So if you'd like to register and sign up for that, that's available through US HUPO registration. But thank you so much again. This was really very fun. I enjoyed it. And yeah, any last words from you?
I'd like to put in a plug for the Mass Spectrometry Imaging Society. We're really focused on letting people have this hands-on experience because you don't always get the some of the aspects when you have a written, even a very detailed protocol. So we do have a workshop at Texas going on, hosted by Aaron Seeley. That's in October. And then the Medical University of South Carolina group is doing a glycomics imaging workshop, which is fairly full, but we'll host another one again. And we also have people down to our site This week we have people from Mayo who are visiting to learn the glycomics and protein imaging workflow. So we really invite you to reach out to the community through the Imaging MS Society website. We would love to interact with people and help answer questions.
Yeah, this is great. I mean, it's so fascinating because you get spatial information, right? So that's preserved. I think that's really special. And yeah, so you showed this very nicely. And yeah, thank you both again. I think this was really good and a great start to our new series and activity. All right. Thank you, everybody. Bye.
Thank you so much.